Good evening. We now move on to our another web lecture series, and it's a pleasure that I have with me today. I know somebody from as long as 2002, so it's a big relationship. It's a 18 years relationship with this lady. And I, as the head of the department, Dr. Malavika Bhattacharya, welcome Dr. Suhnrita Chaudhuri to our department to deliver a talk today. So I'll be just giving a very brief introduction about her. Suhnrita completed her post-graduation from Calcutta University in Physiology and did a doctoral work from School of Tropical Medicine. Her doctoral work was from the year 2009 to 14, where she worked under the joint supervision of Professor Tushar Kanti Ghosh and Professor Suhnrita Chaudhuri, working mainly on the modulation of the TCR signaling pathway upon glioma therapy. After her doctoral work, her postdoctoral life is very illustrious, where she was selected for the very prestigious Fulbright Nehru Postdoctoral Fellow. And being a postdoctoral fellow, she has visited 4 universities. She has worked in 4 universities. From August 2015 to 2016, she worked in the University of Chicago and Northwestern University, that is also in Chicago, working on the role of WNT beta-catenin signaling pathway in T-cell immunology in glioma, that is basically brain tumors. From August 2016 to September 2017, she worked in the University of Illinois at Chicago, working around glioma-induced immunosuppression. And currently she is working at the Barts Cancer Institute as a senior postdoctoral researcher fellow, working mainly on the tumor microenvironment in a pancreatic tumor model. And her publications during this tenure, it's a brief tenure of this many years, she has 26 peer-reviewed international publications, notable among which are the Asteriosclerosis, Thrombosis and Vascular Biology, Pancreatology, Journal of Biological Chemistry, Cell Physiology, Molecular Immunology, Scandinavian Journal of Immunology, Frontiers of Microbiology, which are notable. She has presented her work in 40 national and international conferences, seminars. She is listed in the Who's Who in the World 2019, Who's Who in America 2018. She got the prestigious Fulbright Nehru Postdoctoral Fellowship from 2015 to 2017. She got the Best Paper Presentation Award in the 4th Conference on Respiratory Allergy and Immunology, Best Paper Presentation Award in the Research Workshop on Diagnostic and Therapeutic Immunology. She was also awarded the Senior Research Fellowship by the Department of Science and Technology, DST. Among her academic affiliations, noteworthy are the American Association of Immunologists, North American Vascular Biology, Indian Academy of Neuroscience. So with this, I request Suhnrita to take the stage. And some of her present publications which I was reading through, those who are in science should know that it's really a very envious publication list she has.
Very good evening, everyone. I'm really sorry for the delay. There was some kind of technical glitch at my end. And I thank Malavika Di from the bottom of my heart for such a beautiful, warm introduction. And I've known Malavika Di for a very long time, and she's like a sister to me. And I thank her again for inviting me to give a talk at a wonderful college. But my talk for today will be on the pancreatic cancer microenvironment and what makes this extremely aggressive cancer resistant to therapy. Okay. Right. At the very a few words about the pancreas, the organ itself, which is an important part of the gastrointestinal system, and it is mainly functionally comprised of the exocrine pancreas. And if you look at the histology here, can you see my cursor, everyone? I sure is, well, I hope so.
It's visible. Your cursor is visible.
Okay, perfect. So if you look at the pancreatic histology here, these are the acinar cells, and these are the ductal cells of the pancreas. And these are what comprise the exocrine part of the pancreas. And this exocrine part is chiefly concerned with the secretion of digestive enzymes into the duodenum. And let me tell you that the exocrine component of the pancreas is actually the largest and bulkiest part of the pancreas, and it comprises about 95% of pancreatic mass. If we can magnify the image in this image here, the acinus cells are bigger, and these are the ones that constitute the exocrine part of the pancreas. As for the endocrine part of the pancreas, they are comprised of the islets of Langerhans, which you can see here. And these islets, magnified here, are concerned with hormone production, which regulate blood sugar levels in the blood sugar levels. And these hormones are insulin, glucagon, somatostatin, and the pancreatic polypeptide. So if we explore the anatomical relationship of the pancreas to the surrounding organs and structures, you will see that the head of the pancreas lies in the loop of the duodenum. The body passes behind the stomach distally, and the tail ends up in the hilum of the pancreas. Sorry, the hilum of the spleen. Now, the important thing to notice here is that there is a close proximity, a very, very close proximity, in fact, of the pancreas to a number of important blood vessels in the body, namely the inferior vena cava, the hepatic portal vein and artery, and the abdominal aorta. And this fact actually compounds surgical resection, because that might end up splicing out blood vessels and resulting in huge blood loss. So I want to go into some basics of pancreatic ductal adenocarcinoma. Now, the title of my talk mentions the term pancreatic cancer. Well, pancreatic cancer is actually denotes a number of diseases, a group of diseases, cancerous diseases, the exocrine cancerous diseases of the pancreas, namely the adenosquamous carcinoma, the squamous cell carcinoma, the colloidal cell carcinoma, and finally pancreatic ductal adenocarcinoma. And among the non-exocrine cancers, it is a very rare cancer, and it is worthwhile to mention that pancreatic ductal adenocarcinoma constitutes about 90% or more of the entire number of cases of pancreatic cancer. And unless otherwise mentioned, henceforth, when we talk about pancreatic cancer in this talk, we will be referring to pancreatic ductal adenocarcinoma. Now, a number of precursor lesions precede the development of pancreatic ductal adenocarcinoma. These are termed pancreatic intraepithelial neoplasias. These are usually non-invasive cancers and are very mucinous in nature. And a number of driver mutations are prevalent in pancreatic cancers, and these first reflect in pancreatic intraepithelial neoplasias, abbreviated as PAN-INS. And the first genetic alterations to manifest in stage 1 PAN-IN is a point mutation in the KRAS allele. This is followed by a P16 or P16 loss in PAN-IN. And finally, we can see P53 loss and SMAD4 loss in the PAN-IN3s. Now, patients can remain in the pancreatic intraepithelial neoplasias for a long time, maybe even years, before they proceed into pancreatic ductal adenocarcinomas, abbreviated as PDACs. With the sequential accumulation of mutations, if and when that happens, PAN-INs finally go on to become PDACs, which I must mention are extremely aggressive and fatal cancers, and constitute 3%, they constitute the third most common cause of cancer deaths worldwide. And I need to mention here that PDACs are extremely immunosuppressive cancers, and immunosuppression is one of the key factors that compound therapeutic outcome. And according to the expression of according to the infiltration of immune cells, we can divide PDACs into three distinct phenotypes. The first is the most common. It is called the immune escape phenotype, and it is characterized by a comparative absolute scarcity of cells, namely the CD8 positive and CD4 positive T cells. And it is also characterized by a large preponderance of FOXP3 positive T regulatory cells, which are extremely immunosuppressive in nature. Also, the M2 macrophages, which constitute the immunosuppressive component of the tumor infiltrating, the tumor associated macrophages, and also the neutrophils of the N2 phenotype, which are also immunosuppressive in nature. This phenotype is also associated with a high grade of EMT-like tumor budding, and as can be expected, is associated with extremely poor prognosis. This is by far the most common phenotype of PDACs and the one with the worst prognosis. The immune-rich phenotype, as the name suggests, comprises of a tumor microenvironment with high anti-tumor immune cell infiltration. So there are a large number of effector T cells in the tumor microenvironment, and it excludes pro-tumorigenic immune cells such as the Tregs, the M2 macrophages, the MDSCs, etc. This phenotype, as can be expected, is associated with a better prognosis. And also there is infrequent EMT-like tumor budding here. The third phenotype is very strange in the fact that although it has high infiltration of immune cells, rather than being able to fight off the cancer, it is actually poorly immunogenic due to the existence of high-grade EMT-like tumor budding here, and this also has poor outcomes for patients with a high expression of PDL1. Now, a lot of people know that the current standards of care for PDACs include surgical resection. As I've mentioned before, surgical resection is complicated by the fact that pancreas lies in close proximity to major blood vessels, and also due to the fact that PDACs are most often detected at very late stages when the cancer had metastasized, and this further compounds surgical resection. It also, one of the other current standards of care for PDAC is also radiotherapy, but sadly enough, radiotherapy has very poor, is not really very effective in PDAC patients, as PDAC patients are famously extremely radio-resistant. So that leaves us with chemotherapy, and although chemotherapy is still the current standard of care, it still does not have very significantly improved therapeutic outcomes. Gemcitabine has been in use for quite some time now. It only moderately improves survival. FOLFIRINOX is a newer chemotherapeutic drug, and it is a 4-drug polychemotherapeutic regimen comprising of folinic acid. You can see that it is an abbreviated name. I'm sorry, the name FOLFIRINOX is actually an abbreviation for the components of the polychemotherapeutic regimen. So there is folinic acid, there is 5-fluorouracil, there is irinotecan, and oxaliplatin. The newest entrant in the chemotherapeutic drug regimens for PDAC is nab-paclitaxel in combination with gemcitabine. So nab-paclitaxel is a nanoparticle albumin complexed with paclitaxel, and it is administered in combination with gemcitabine. This improves therapeutic outcome and survival somewhat, and patients have moderately improved quality of life. However, none of these current standards of care have significantly done anything to improve the status of PDAC patients anywhere in the world. This is very well illustrated if we look at this chart here, which illustrates the comparative cancer survival over time. And what is extremely sad to look at is that pancreatic cancer has the worst 10-year survival, the worst 5-year survival, and even the worst 1-year survival of all the cancers listed here. That is despite the fact that there are some very aggressive cancers on this list, including brain cancers and lung cancers. The story is still very abysmal because if we look at changes in survival from the 1970s right up to the 2010s, pancreatic cancer patient survival still remains as abysmal as it was in the 1970s. More and more research is required into improving the therapeutic outcome of patients with pancreatic cancer. Now, the tumor microenvironment of pancreatic cancers are a major component which compound therapeutic outcome. The tumor microenvironment comprises the tumor stroma, which actually is far more bulky than the actual tumor itself, comprising up to about 80% of the tumor mass. And if we look at the definition of tumor microenvironment in Nature Reviews Cancer, it says that the tumor mass consists not only of a heterogeneous population of cancer cells, but also a variety of resident and infiltrating host cells, secreted factors, and ECM proteins, collectively known as the tumor microenvironment. They further go on to state that tumor progression is profoundly influenced by interactions of cancer cells with their environment that ultimately determines whether the primary tumor is eradicated, whether it metastasizes, or it establishes a dormant micrometastases. The tumor microenvironment can also influence therapeutic outcome and resistance to therapies, thus justifying the recent impetus to target components of the tumor microenvironment. So this gives us an idea of why studies of the tumor microenvironment of pancreatic cancers, and indeed many other cancers, is so important at the laboratory level. Now, one of the important mouse models that are in use today to study the tumor microenvironment is the genetically engineered mouse model called the KPC mouse model. Now, this utilizes the CRUK system to introduce mutations in the KRAS allele. The KRAS allele, I've mentioned before that mutations in the KRAS allele and T53 mutations are commonly observed in pancreatic ductal adenocarcinoma patients. So by the utilization of a CRUK system, these mutations are preferentially introduced into the pancreas of these mice. And these KPC mice have been shown to recapitulate the tumor microenvironment very well. So if you look at the histology here, this is very similar to the histology of PDAC patients, where you see extensive fibrosis in the slide that you see here. And this extensive fibrosis in PDACs is termed as the desmoplastic reaction. The desmoplastic reaction in PDACs is such that it causes extensive barriers to the penetration of immune cells or any other drug delivery and compounds therapeutic outcomes. You also see here that there is an absolute disorientation in pancreatic histological architecture, and you see a number of malignant epithelial cells interspersed with the fibrotic stroma. If you look at the right-hand side panel here, this is an opened up abdomen of the KPC mouse. And this again very well mirrors the picture of a PDAC patient's abdomen, where you see a very stiff fibrotic, big-sized pancreatic tumor. And you also see micrometastases in the liver. Although it's not visible in the picture, micrometastases also occur in the spleen, the diaphragm, and the lymph nodes, and sometimes they even metastasize into the lungs. And needless to say, the KPC mouse has a very bad prognosis. It has cachexia, weight loss, and all the other characteristics that have been associated with PDAC patients. So in effect, the KPC mouse model is a good mouse model for the study of the TME. Now, this is a slide where I talk about the breeding strategy of the KPC mouse, but I won't go into details of that right now. If we have time later, we can discuss that more. But it is to be mentioned that GEMMs, or genetically engineered mouse models, are extremely expensive to use, and they are also time consuming in the development of cancer. So a lot of labs, including my own, including the work I do, I use KPC derived mouse PDAC cell lines. The cell line I use is called TB3T043. And I use this to develop a syngeneic mouse model by orthotopic injection of mouse PDAC cells directly into the pancreas of the mouse. And this is done against a C57 black 6 background. And what I need to mention here is that after the PDAC cells are implanted orthotopically, we image these mice via MRI every 5 days to assess the tumor growth and the tumor size. And in the UK, where I work, there are very strict limitations on the size up to which the tumor is allowed to grow so as to minimize pain and discomfort to the animal. So right now, that limit has been set at 100 millimeters cubed in diameter across the tumor. So we monitor the tumor growth so that as it approaches the 100 millimeter cubed mark, we sacrifice these animals and take them for downstream analysis. This is only one of the many animal models which are in use for the study of PDACs. However, this is the pancreatic model that I use, and it has given me some very good results so far. So this is some data that I've generated in my lab. If you look at the left-hand side panel here, this is an abdominal MRI of the orthotypically induced mouse PDAC that I've talked about just now. And you'll appreciate that this is the whole abdomen of the mouse. And looking at this circular-ish structure here, this is the pancreatic ductal adenocarcinoma of the mouse. And relative to the size of the abdomen, you will realize just how huge this has become. And this happens in a span of about 30 days. So the cancer growth is very fast and very aggressive. And this reflects in the histology of the mouse PDAC. This is one of my histological slides. And you can see here that, as I had shown in the KPC mouse model, again, there is extensive fibrosis here, huge numbers of malignant epithelial cells. And the interesting thing to notice here is that there is a change from the typical cuboidal epithelium of the normal pancreas to a columnar phenotype, and these columnar phenotypical cells, epithelial malignant cells, are seen to line the ductal cells here. And these are some of the typical features of the mouse PDAC in my animal model. And this was found to be a good animal model for the study of PDACs and TME of PDACs, because this efficiently recapitulates what happens in PDAC patients. So if I'm allowed to talk a little bit about what happens in the pancreatic microenvironment, the changes that occur during carcinogenesis, let me start by telling you that the normal pancreas is comprised of resident fibroblasts, which are inactive fibroblasts, not cancer-associated fibroblasts. It also has quiescent pancreatic stellate cells, abbreviated as PSCs, immune and vascular cells, which crosstalk amongst each other to play critical roles in wound healing and tissue repair. However, in response to oncogenic stimulation or pancreatic tissue injury, we can see that carcinogenesis ensues, and this disrupts the normal communications between the PSCs and the resident fibroblasts. And the resident fibroblasts and the PSCs are two of the chief cell types in the pancreatic cancer patients, which have a huge role to play in the TME development and maintenance. And these two cells together compound therapeutic outcome. So now the normal communications between the PSCs and the PFs, which have become the cancer-associated fibroblasts or the CAFs, with the immune cells and the vascular cells are hampered, and this gives rise to a favorable microenvironment for cancer progression. Cytokines and growth factors, which are secreted by the tumor cells themselves, activate these PSCs and the fibroblasts into activated PSCs and CAFs, and these play key roles in development and maintenance of stromal architecture and the desmoplastic reaction. In the lowest panel here, you see intense fibrotic reaction in place, and this is characterized by activated PSCs and CAFs, and these two cell types now produce alpha SMA, and these, in conjunction with the vascular elements and the immune cells and acellular components such as collagen, hyaluronan, fibronectin, etc., and cytokines and growth factors, are now stored in the ECM here. So in this stage of the disease, the myeloid-derived suppressor cells, which are called MDSCs, the M2 macrophages, the N2 macrophages, T regulatory cells, all of which are immunosuppressive immune cell phenotypes, induce a pro-tumor phenotype into the cell, into the PDAC. Now, the characteristics of PDAC that compound therapeutic outcome majorly involve abundant stromal content. So the huge desmoplasia that we've spoken about in the previous slides is an effective barrier to the penetration of any drugs that are to migrate into the parenchyma of the tumor. There is also a very notable thing to mention, that unlike many other tumors which have rich blood supply, PDACs are notorious for being very poorly vascular. And being poorly vascular, they are also poorly pervasive, poorly perfusive to drug delivery. And this also compounds therapeutic outcome. And of course, tumor-induced immunosuppression provides a further barrier towards therapeutic outcome. So in effect, the tumor microenvironment acts as a pharmacological barrier to compound therapeutic outcome of PDAC patients. So this is a picture that I've taken from the Nature Reviews Cancer recent paper. And this panel on the left is a mouse PDAC histological slide. And this again is a human PDAC histological slide. So in both of these, if you can look at the black arrows, you see an intense desmoplastic reaction. And the white arrows here denote the malignant epithelia that is associated with PDACs. Now, what really happens in the desmoplastic reaction is that fibroblasts associate very tightly in conjunction with the cancer cells, and the acellular components such as collagen, hyaluronic acid, fibronectin, etc., also deposit in the ECM. Hyaluronic acid is famous for trapping water molecules and increasing osmotic pressure inside of the inside of the tumor parenchyma, so as to impede drug delivery, along with the fact that the physical barrier presented by the fibrotic network is also huge and provides for a very important barrier to drug delivery. Now, I'm going to show you a few slides from my own PDAC model. So this is serous red staining, indicating that serous red stains for collagen in the pancreas, in the tissues. And this panel on the left here shows the normal mouse pancreas. And if you see the relative scarcity of red staining in the left panel, you'd appreciate that the normal mouse pancreas has very scarce amounts of collagen. Now, you will be absolutely baffled when you look at the mouse PDAC slide here, and the huge preponderance of red staining in this slide shows just how aggressive the collagen deposition in the ECM is in my mouse PDAC model. These are some other slides that I've presented from my data, and this will also show you how I wish I could actually magnify these images, but I'll try and do, I can magnify them, that's wonderful. So, oops, I can't pull them down. Well, I'll do the best I can. So Ki67, so these are all TME markers for PDAC. Ki67 is a very important proliferation marker. And in human PDACs, Ki67 scores, higher Ki67 scores, are associated with the worst outcomes. So if you see my section here, let me see if I can, yeah, I probably can, oops, no, this is the best I can do. Well, if you look at the Ki67 staining here, if you look closely enough, you'll see a large preponderance of brown staining here, denoting Ki67. So you appreciate how proliferative these PDAC cells are, and also how aggressive and how horrible the prognosis is for the mice with these PDACs. Endomucin stains for endothelial cells that line the blood vessels, the inner walls of the blood vessels. And if you look at the endothelial, at the endomucin staining, so these are immunohistochemical staining, and are stained with DAB, which leaves brown deposit. So if you look at the scarce brown deposits in my endomucin staining slide, you'll understand how scarce the blood vessel distribution is, and even where they are present, the perfusion status of the blood vessels is very poor. Okay. Alpha SMA, as I may have mentioned before, is secreted by activated fibroblasts, so the cancer-associated fibroblasts or CAFs, and also by the activated pancreatic stellate cells or PSCs. And this denotes the extent of fibrosis in the PDACs. And in my mouse model, you'll see that alpha SMA staining is very dense, denoting the high activation status of the CAFs and the PSCs, as well as extensive fibrosis in my sections. The section here on the lower left denotes CD3 staining. Now, CD3 staining is, CD3, as we all know, is an important marker for T cells. And if you look at the very low amount of CD3 staining here, you'll understand how low infiltration of tumors, of the anti-tumor infiltrating T cells are. Also in the panel in the middle, in the lower middle, is F480 staining. F480 stains for macrophages. The high staining of F480 here probably shows that there is a high preponderance of the pro-tumorigenic M2 macrophages, but this is just a conjecture because I haven't actually looked at, I haven't actually used differential markers for the M1 and M2 macrophages here, but I plan to do that when I go back to the lab. But I'm guessing that since there is such a big preponderance of M2 macrophages in the PDAC of human patients, the high F480 staining here could be adequate representation of the preponderance of M2 macrophages in the TME. And a very notable feature of PDACs is that they are profoundly hypoxic. And this hypoxia is very contraindicatory to the therapeutic outcome. Carbonic anhydrase 9 is an important marker for hypoxia, and the hugely brown, aggressive staining here for CA9 will show you just how aggressively hypoxic the conditions are in the tumor microenvironment in my PDAC mouse model. Now I'm going to try and zoom out so we can go to the next slide. Yes. Okay. So chiefly, there are two major components of the PDAC TME. The cellular component, which comprises the CAFs, the PSCs, which I mentioned before are very important cell types of the TME, concerned with the maintenance and development and maintenance of the desmoplastic reaction. It also has the vascular cells and the inflammatory immune cells. The acellular component comprises of collagens, in this case collagen 1, 3, and 4. It also has periostin, fibronectin, and hyaluronic acid. Now, without actually going into this extremely wordy slide, I'm just going to tell you that the chief cellular components of the PDAC TME are CAFs, the CAFs, the activated PSCs, which are responsible for the desmoplastic reaction and maintenance of the desmoplastic reaction. They also signal to immune cells and cause profound immunosuppression, and the activation of these cells is dependent on oncogenic signals from the PDAC cells themselves. The bone marrow derived cells are another cell type which is important in PDACs. These are recruited to the stroma quite early in carcinogenesis, and these are concerned with the progression of carcinogenesis, and also these are responsible for activation of the myeloid-derived suppressor cells and mast cells, which cause immunosuppression in the PDACs. The MDSCs are responsible for the suppression of T cell activation by TGF-beta secretion from the PDAC cells themselves. And these are actually immature myeloid cells, which are activated into MDSCs by oncogenic signals from the PDAC cells themselves. And high levels of high levels of these MDSCs have been known to lead to premetastatic niches, tumor invasiveness, angiogenesis, and horrible prognosis of the PDACs. TAMs are the tumor-associated macrophages, and the large preponderance of TAMs in the TME is that of the M2 macrophages, which are pro-tumorigenic and immune suppressive. So these M2 macrophages are also known to cause T cell depletion and T cell deletion in the tumor microenvironment. T cells. So of the variety of T cells.
Suhnrita, can I interrupt? Suhnrita?
Yes, of course. Yes, yes.
What I'm saying is that let's stop in this particular slide, okay? Because there is 3 minutes left. I'm disconnecting this session and I'm sending a fresh link. It will take 5 more minutes.
Sure, sure. Where will you be sending this link, Malavika?
I'll send you to your WhatsApp.
Okay, cool, cool.
Okay.
No worries.
So I'm disconnecting it now and I'm sending the link. It will take 5 minutes, okay?
Sure, sure, sure. No
we'll
problem.
start with this slide, okay?
Okay, sure. Derived IL-18 to promote immune tolerance by the PD-1, PD-L1 pathway. There's also a role for microvasculature. And among the acellular components of the PDAC TME, the most important members are collagens, hyaluronic acid, periostin, and fibronectin. Now, the collagens are very important in the sense that these are deposited and they contribute to the largest part of the stromal barrier formation in PDACs. And this influenced not only drug resistance, but also very poor prognosis. The hyaluronic acid that is also deposited in the PDAC TME um, binds and traps to water molecules. I mentioned this before, and this causes a high pressure on the neighboring structures, as well as elevated interstitial fluid pressure, or IFP, and high osmotic pressure within the tumor. And periostin is expressed at a 42-fold higher rate than in normal pancreas, and it also has been shown to promote PDAC cell invasiveness, hypoxia, resistance to hypoxia-induced death, and epithelial to mesenchymal transition. Fibronectins are also expressed in high levels, and these are secreted by calves, along with collagens, cause anisotropic fiber orientation, which influences cancer cell migration. Now, the chief cytokines that influence therapeutic outcome in PDACs include TGF-beta, IL-10, IL-6, IL-1 beta, and IL-17 and TNF-alpha. Now, TGF-beta has a pleiotropic role in PDACs, in the sense that in the early stages, it has been shown to actually inhibit cell cycle. So cell cycle progression in PDAC cells. So it is actually anti-tumor, it actually has anti-tumor properties in the early stages of PDAC carcinogenesis. However, as the disease progresses, this TGF-beta enhances invasion and metastasis by inducing epithelial to mesenchymal transition in the advanced stages. IL-10 is also an important cytokine which is secreted from the M2 macrophages, Tregs, mast cells, and indeed by the PDAC cells themselves. And it is associated with the recruitment of M2 phenotype macrophages into the tumor milieu, as well as EMT-induced, EMT, which is induced in early tumorigenesis. IL-6 is another important cytokine which promotes angiogenesis and also causes cancer cell migration and EMT. IL-1 beta promotes cancer growth, invasion, and metastases. IL-17 is secreted by another population of the T cells known as the Th17 CD4 positive cells, and this plays a major role in the recruitment of MDSCs into the tumor milieu, and this causes a stem-like phenotype of the PDAC cells, and it also causes tumor initiation and progression, but partial EMT only, which is actually a significant feature of cancer cells, and this depends on oncogenic KRAS mutations. TNF-alpha is another important cytokine secreted in the PDAC milieu, and this is usually secreted by the M1 macrophages, neutrophils, mast cells, and the stellate cells. It antagonizes M2 polarization and is associated with PDAC initiation. It promotes angiogenesis and metastases by activating the NF-kappa B signaling pathway. Now, this in this slide, we will explore a little bit the relationship between PDAC cells, here, the fibroblasts, here, and the ECM. So what happens is the PDAC cells via oncogenic stimuli recruit the fibroblasts and convert them into CAFs in the early developmental stages. And this is done by the secretion of the sonic hedgehog, SDF1, TGF-beta, and PDGF. Fibroblasts then deposit extracellular matrix components by secreting PDGF and SDF1. And this dense ECM now promotes PDAC development through the stimulation of integrin and fat pathways in the PDAC cells, and it also causes contact-mediated lymphocyte trapping. So here we go into a little bit of detail about the crosstalk between the PDAC cells, the TME, the immune system, and potential therapeutic agents that can be used to target the TME. Now, we can see here that the TME components play a role. Sorry, the TME components interplay with PDAC cells. You can see that they're shown by, the interplay is shown by the black arrows here. And this interplay occurs through the secretion and production of cytokines such as TGF-beta and soluble factors and growth factors such as the PDGF, FGF2, etc. And this leads to an increase in tumor proliferation, tumor growth, and metastases. And PDAC cells also cause an activation of these PSCs and CAFs, which I mentioned before. And these again crosstalk with immune cells and also deposit increased amounts of ECM components. The continued crosstalk that exists between the TME and the immune infiltrates cause T cell depletion. So the T cells here express PD-1, and the overexpression of PDL1 by the PDAC cells themselves cause a deletion of the effector T cells from the tumor milieu. And among the several molecules that target the different mechanistic outcomes of PDAC, you see several green boxes here. So these boxes include the TME targeting, the possible TME targeting agents, which can improve therapeutic outcome by targeting the TME. Among these, nivolumab, CCX872B, the gamma delta T cells, etc., are important to mention. Interactions, this is another slide explaining briefly about the interactions between the immune system, the TME factors, and the immune cells. You can see that the gamma delta T cells promote tumor development and block the function of effector CD8 and CD4 T cells. And fibroblasts actually recruit B regs, as I mentioned before, by secreting CXCL13. And the secretion of IL-35 by the B regs also results in tumor progression. Another important mechanistic feature of PDACs is increased hypoxia. And this results from the extremely abundant stromal content in the PDACs, which leads to high interstitial fluid pressure, IFP, leading to hypoxia and the stabilization of HIF-1 alpha. And the stabilization of HIF-1 alpha now leads to increased glycolysis and glucose uptake. So there is a shift from metabolism in the PDAC metabolism from oxidative phosphorylation to glycolysis and glucose uptake. This results in acidification of the extracellular environment by lactate production. So the combination of hypoxia, the acidic milieu, and high IFP in the TME, these further drive tumorigenesis and tumor progression and chemoresistance. The TME adapts this altered metabolism and causes increased gluconeogenesis, where the PDAC cells consume lipids and proteins. An invasive EMT and a metastatic phenotype ensues. The PDAC immune cells interact with TME components, resulting in the inactivation of the cytotoxic antitumoral response. And the TME could now influence treatment efficacy through different mechanisms, including drug delivery, modulation, immunosuppression, vascular remodeling, metabolic activities, and signaling pathways involved in DNA repair and apoptosis. Now, a number of factors I've mentioned before contribute to therapeutic resistance in PDAC. Factors contributing to radioresistance include mutations and altered signaling pathways in the PDAC cells, which have roles in radiation resistance by affecting DNA damage repair, sensing of DNA damage, cell cycle of the cell. And these days, potential radiosensitizers, including 5-fluorouracil, gemcitabine, etc., are being used increasingly to improve radiosensitivity. with varying degrees of therapeutic outcome. And a number of synthetic drugs are now being tested for radiosensitizing and radioprotecting pancreatic ductal carcinoma patients. These are only in preclinical study stages. So a number of major factors contribute to the development of chemoresistance in pancreatic cancer. The major players are PSCs, tumor initiating cells, also called cancer stem cells, the cancer associated fibroblasts, and the microvesicles. So in sorry, কিছু বললেন?
না কিছু বলিনি রে।
আচ্ছা, okay. So what happens is the PSCs are, I've mentioned before, important stromal components in the PDAC, and these are initially activated in the regions surrounding defective cells of the pancreas, and these are encapsulated to prevent the damage from spreading to other parts of the pancreas. However, upon oncogenic signals, these same PSCs, upon activation from oncogenic stimuli from the PDAC cells themselves, initiate hypoxia and fibrosis within the stroma and create the so-called desmoplastic barrier, which compounds therapeutic outcome and is largely responsible for chemoresistance. Upon activation, these PSCs also acquire a myofibroblast phenotype. So the cancer-associated fibroblasts, the CAFs, are also activated, and together these two cells deposit into the ECM, alpha SMA positive cells. These together deposit into the ECM and compound therapeutic outcome by being chemo-resisting. The factors contributing to activation of quiescent PSCs into active PSCs include tumor-derived TNF-alpha, TGF-beta, IL-1, IL-6, IL-10, and also PDGF. Coming to the tumor-initiating cells, there are, within the tumor themselves, there are CD133 positive stem-like cells, which are cancer stem cells, and these have increased expression of the ADC transporters and the anti-apoptotic genes such as BCL2, and these confer chemoresistance in the PDACs. Also, another important player in chemoresistance are the PDAC cell-derived microvesicles, which play a main part in the elimination of chemotherapeutic drugs such as gemcitabine from both the intracellular space as well as from the TME. Sorry. Okay. So going to the next slide, these days, CAR T cells, which are chimeric antigen receptor T cells, which are adoptive T cell therapy in PDACs, are more and more being tested for therapy of TME. However, CAR T cell penetration into the cancer is again impacted by the dense desmoplastic TME of the PDAC. So CAR T cells struggle to penetrate into the dense desmoplastic stroma and fail to traffic to the tumor site. The Treg cells that release immunosuppressive signals directly kill and ablate the CAR T cells from the tumor microenvironment. And MDSCs within the TME release anti-inflammatory signals and mediators that reduce the expression of CD3 zeta expression in the CAR T cells. They also, cancer cells, the PDAC cells themselves, also release the FAS ligand to promote CAR T cell apoptosis. And HIF1 alpha and lactic acid released by the tumor cells create a hypoxic microenvironment which reduce CAR T cell effector function. This picture is actually taken from one of our own publications in Pancreatology this year. So among the possible therapeutic drug targets that have now begun to be tested in labs all across the world, hyaluronic acid inhibitors, ROCK inhibitors, and the activated FAC inhibitor have been shown to have preclinical efficacy. So when we talk about hyaluronic acid, the degradation of hyaluronic acid by the small molecule inhibitor PEG-PH20 decreases the intratumoral pressure and improves chemotherapeutic delivery within the PDAC genetically engineered mouse models. The activated Rho kinase, give me 1 second, please. Sorry about that. Yeah, so the activated Rho kinase in PDAC causes ECM remodeling through through increased production of MMPs, and this causes metastases. Also, microvesicle density is lowered, and this causes a significantly decreased outcome of chemotherapeutic drug delivery within the tumor. So in some labs, they've tested a small molecule inhibitor of activated ROCK, which is called Pacidil, and this has, in preclinical settings, shown to improve therapeutic outcomes. Another small molecule inhibitor of activated FAC, which actually causes lower CD8 T-cell infiltration and desmoplasia, and causes lower chemotherapeutic drug delivery. So when the activated FAC is inhibited by defactinib, these therapeutic outcomes again have been seen to be improved in the therapeutic setting. We now move on to the vascular normalization as a therapeutic strategy in PDACs. I've mentioned before that PDACs are notoriously avascular, and even when the few blood vessels that exist occur, they are very poorly perfusive, so they inhibit drug delivery. So this poor PDAC vascularization has decreased pericytic coverage, decreased tumor cell perfusion, decreased O2 tension, and consequently decreased chemotherapeutic drug delivery, and has higher immunosuppressive macrophages. Now, normalization of the vascular infrastructure in the PDACs by anti-VEGF-VEGFR axis improves pericytic coverage, tumor perfusion, O2 tension, and consequently chemotherapeutic delivery. So this is now a selected list of ongoing clinical trials that are targeting the PDAC stroma. Among these are inhibitors for FAC, inhibitors for the IL-1 receptor, PDGFR alpha, CXCR4, CSF1R, IL-6 receptor, IGF-1R, etc., etc. And it remains to be seen how targeting the PDAC stroma by these several TME modulating molecules has any effect on the survival and quality of life of PDAC patients. So in conclusion, we can say that the PDAC TME is a tumor-promoting milieu comprising of cancer-associated non-malignant cells and acellular factors that promote tumor proliferation, immune evasion, metastases, and therapeutic resistance. From the talk, the take-home message is that drugs that target one or more factors in the PDAC TME may impede several aspects of tumor growth and invasion, and thus combinatorial treatment using agents that target tumor cells along with agents that target the TME may improve prognosis and provide survival benefit for PDAC patients. Now, at the very onset, I thank Malavika Di for this very kind invitation. I will also thank my PI, Professor John Marshall, who is an extremely kind individual, and my team members at BCI. And this is a picture of the entrance to my campus. Uh, in this picture, you can see a very blue sky, which I must mention is a rare feature in London because London skies are notoriously gloomy and rainy. This is actually, if you can look at the window here, this is actually a better representation of a London sky. See how gloomy it is? And from this side, we have now established that I am the shortest person in the group. And yeah, so I proceed to thank my funding agencies, PCUK and CRUK. Thank you. Thank you for your kind attention, and thank you for dealing with the delays caused by the technical glitches. Thank you very much for your attention.